అయితే నేను వెబినార్ just 5 minutes. Okay. So they may pick up and transfer certain genes of the bacterial chromosomes. They may affect certain characteristics of the bacterial cells. The plasmid differ from the viruses in following two ways. They do not cause damage to the cell generally or beneficial. That not the effect of the bacterial chromosomes, not effect of the initial organelles of the bacteria. Only beneficial. Beneficial means functionless of the nucleus. It can functioning of the also the take functioning of the plasmids. They do not have extracellular forms and exist inside cells simply as a free and a typically circular DNA. So this is also then the major important of the properties. It is also the several type of the properties. Then mainly 9 properties are there. They do not have extracellular forms and exist inside cells simply as a free and typically circular part. So this is also then incapability of plasmids. So what are the capability and incapability? Capable do it. Then incapability does not do. Then in some cases, a single bacterial cell contains several different types of the plasmids. So Borrelia, Burdovely that causes Lyme disease. For convenience passes 17 different circular and linear plasmids are there. So that the type of the disease in a condition when a plasmid is transferred to a new bacterial cell that already passes another plasmids. And based on the disease qualities of the plasmids are exhibiting the diseases of the plasmid, it can transfer to the other bacteria that cannot affect to the own bacteria or parental bacteria. It is a commonly observed that the second transferred plasmid is not accommodated and it is lost during the subsequent replication. This is important. Means incapability quality. What is the incapability quality of the plasmid? The second transferred plasmid is not accommodated and is lost during the substance replication. This condition is called the plasmid incapability. That is also measuring but 3 lines. And 2 plasmids are said to be incapable. Means incapable means does not capable means doing the one function. A number of incapability groups of plasmids have been recognized in bacteria. Then a large groups of number of bacteria, incapable bacteria, capable bacteria. The incapability quality depends on the plasmids of that own bacterial cell. The plasmids of one incapability group excluded each other from replicating in the cell, but generally coexist with a plasmids from other groups. The plasmids of a incapability group share a common mechanism of regarding their replication and are thus related to the one another. Therefore, all through bacterial cell may possess a various types of plasmids, each of the genetically distinct. Then the genetical distinct qualities are there. That is called based on the capability quality of the plasmid, incapability base quality of the plasmids. That is also, it is a classification of the based on a function plasmids and a classification. F plasmid, fertility plasmids, R plasmid, resistancy plasmid, COL plasmids means it is also causing of the containing genes that codes for bacteria. It proteins that can kill bacteria. Cold means kill bacteria. Then based on the only 3, additionally to the total 5 type of the plasmids are may be classified that based on their function and also called a quality or the character. So these are examples are PBR 322 artificial, PBR 327, PBR 325, PBR 328, PUC, plasmid University of California 8, PUC, plasmid University of California 9. It's a 12. also 13. So this is also called additional, also called artificial plasmids, which can, the types of the plasmids, various types of the plasmids naturally occurs in bacterial cells. And the most favorable classification of such plasmid is based on their functions encoded their genes. Two type of the character based to the plasmids, which can classify the 5 types. First one, that is called F plasmid. The following are the main types of plasmids recognized based on the above mentioned characters. Above mentioned means 2 characters. First character is also called based on the, and you say their main function. Second one, encoded their genes. Encoded their genes. F plasmid, F means that is also called F stands fertility quality. The fertility is based to conjugation of the bacteria. That the F plasmid is F factor is a very well characterized plasmid. It plays a major role in conjugation in bacteria, also called Escherichia coli bacteria, and was the first to be described. It is the plasmid that confers maleness on the bacterial cells. The term sex factor is also used in the refer to the F plasmid. Sex factor or F factor, both are same. F means fertility, then sex means also quality of the reproduction or quality of the sexual reproduction. Because of its their property, F plasmid is a circular double stranded DNA molecules of 99